I have had many requests. I have brought my family before to past SSE meetings, and I have had, no doubt, I would say about 24 requests from people asking me, where's my wife? Where are my kids? And so I got on the phone. I called up my wife, and I said, I think you're going to need to send a picture. She goes, I'll send the beach picture. I said, okay, that'll be fine. I'll put it at the front. So therefore, here they are. There's my son Gregory, 5 years old, in the middle hiding. There's Benedict being held by my wife Karen, and there's Lydia at 1.5, whom I'm holding right there. I am from Florida, for those of you who are not aware, so this is at the beach. And for those of you who are from Colorado, this is called an ocean behind me. It's a big body of water. So we've got lots of beaches there, and so I just wanted to share that with you as at the beginning of the talk. Now, let's see, which one? There we go. The talk, Challenging the Insect Olfaction Paradigm, New Evidence. Some of you probably knew that I was going to be talking about insect olfaction. This is specifically what we study more so than anything else at the laboratory, although we do study other parts of bioelectromagnetics. But having the background in entomology, this has really forced me in order to go after the insects more so. And it was a number of years ago that I realized that there were problems, as many of us have found in certain paradigms, and the insect olfaction paradigm was one of them. Now, I did present in 2009, and in 2009 I talked about the temporal evidence against the current theory of insect olfaction. However, now, 3 years later, there is some new evidence to report. Therefore, I'm going to first introduce what the insect olfaction paradigm is, so that all of you are all up on the same boat and you're understanding where I'm coming from. And then I'm going to introduce the new evidence so that you can see and follow along why I have some difficulties with that particular paradigm. Olfaction in insects. How is it accomplished? Most of us know that insects smell with their antennae. We do know that instinctively. So we've got a large bipectinate antennae from the Saturniid moth off to the left. We have another clear-winged moth off to the right, but you can see the antennae are very prominent. And that is how almost all insects smell. Now, there is some smelling and tasting on the feet and the wings and even on the back end of the insect, but for the most part, odorants are picked up by the antennae. Now, if we zoom in on the antennae, we can see that there are some small structures, and I want to point those out because it's not the antenna proper that is necessarily involved in insect olfaction. We have these very tiny sensilla, sometimes called hairs. Sometimes they're called trichoid sensilla, which means a hair sensilla. And these are the actual structures which are involved in insect olfaction. This is what they're actually using to detect the odorants, in some case, a pheromone. So I've zoomed in on just one of these hairs, and these things are microscopic. You can't see them with the naked eye. But if we zoom in on this, we see these are the cells underneath. There's a trichogen, tormogen cell, thecogen cell, 3 cells that are involved in the formation of the hair in the socket. Up above, we have something sticking up above the cuticle, which is the hair-like portion that you see right there. There are some holes in the top. These pores are very tiny, somewhere on the order of 10 to 50 nanometers. Can't see those either. And the pheromone, in this case the long-chain carbon molecules, which is indicative of most lepidopteran pheromones, those being the butterflies and the moths, would then enter the holes and make their way through this watery matrix right here and actually impinge upon a dendrite. So there's a dendrite extending up into the hair right there, which is the final detector of the insect pheromone. What I'm going to do is I'm going to zoom in right now. So I'm zooming in on just this part. So I don't want to talk about the entire hair, but I do want to focus on the pores, the outside cuticle, and the dendrites on the inside. Let's continue. This is now the close-up that I just described to you with that red rectangular box. Now, we can see the outside cuticle. Those blue dots are what I'm going to call the pheromone. These yellow dots are the pheromone-degrading enzymes. They eat up the pheromone first chance they get. They're very good at it, and they are found on the outside. In addition, they're also found on the inside, so any pheromone that's going to get inside is also going to get picked off by the pheromone-degrading enzymes, and they're working very, very hard in order to eat the pheromone as quickly as possible. Now, what happens is because the pheromone cannot cross this watery matrix, because it is a long-chain fatty acid, oil and water don't mix, therefore it has to pick off a ride from another molecule. This other molecule is called a binding protein. This binding protein picks up the pheromone at the pore, just like that. It travels across via diffusion, makes its way to the dimer receptor. We actually know that the receptor is composed of 2 proteins, not just 1. And this dimer receptor then somehow is able to pick and recognize the pheromone or the pheromone binding protein complex. It is not quite clear whether or not there is a separation of the pheromone or whether or not they actually are detected in the complex. And this separation, if it does occur, occurs at a pH of 5. Otherwise, there will be no breaking of the bond between the binding protein and the pheromone. So just to show you what the relative size is of the binding protein versus the pheromone, the binding protein is much bigger. It's about 66 times bigger, assuming a volumetric mass of a sphere. And they're not spheres, I understand that. The pheromone binding protein is roughly globular. The pheromone is not usually circular. But I actually drew this to scale so that you can see the size difference between them, and that if the pheromone does not escape, remember that part of it is going to be bound to the binding protein and will not be able to be necessarily detected. So we've got a dilemma right here. Is the pheromone alone being detected at the dendritic membrane, or is the pheromone binding protein complex? In other words, it doesn't break up. There are 2 fields of thought. I know a number of American researchers who feel the first. I've talked to German researchers who believe in the second. But if the receptor detects the pheromone shape, then obviously the pheromone alone needs to do that. However, we have a different situation if the Germans are correct. The Germans are correct, and that the complex then binds with it. The receptor detects the whole complex and identifies the pheromone based on this complex. This is the summary slide that I gave back in 2009. I wanted to jog your memory for those of you who were there with me in 2009. I had expressed that the temporal evidence did show that it's actually impossible for the pheromone or any odorant in order to reach the dendritic membrane in time in order to explain insect olfaction. Time to adhere to the sencilla, 0 milliseconds. Time to diffuse through the wax layer takes between minutes and hours. Time to diffuse through the pores, nothing's been published, it's unknown. Time to actually bind the pheromone, only about 1 millisecond. Time to transport the pheromone, only if it's a perfect watery matrix. Diffusion laws say that it's going to take a minimum of 12 milliseconds. Time to dissociate from the pheromone, 9 milliseconds if a pH of 5, 100 seconds if we don't have that pH of 5, and finally time to activate the receptor, less than 1 millisecond. It's actually on the order of picoseconds. So if you add this all up, you can see that it's impossible to reach the goal of 1 to 10 milliseconds. Why is it 1 to 10 milliseconds? Because we know the insect can smell within about 1 to 10 milliseconds. So if you cannot explain how the pheromone gets there, then how is detection occurring? And we know very reliably that it's occurring in less than 10 milliseconds. For you biologists, I want to put this into perspective. For you biologists, or maybe neurobiologists, I know there's a couple of you in the audience right now. The synapse. If we take a look at the time course of events of a neurotransmitter as it goes across the synapse, it takes between 3.3 and 5.3 milliseconds for synaptic transmission to occur, for the process of that to occur. The insect pheromone is on the same order of detection on a time scale as a synapse. The synapse, and for you neurobiologists, it would be a type 2 acetylcholine receptor. The synapse is running about 20 nanometers. The insect sencilla, the pheromone, has to travel between 1150 to 2000 nanometers. So there's a very big difference in space that needs to be traveled. And therefore, to assume that the synapse and the sencilla are smelling at the same time is hard scientifically to believe. So what else is going on? Some of you know the direction that I'm heading in right now because obviously I'm involved in bioelectromagnetics. So is there a possibility that there is something else going on besides the classic diffusion, lock and key method, which is talked about in the textbooks and in the insect physiology courses that are taught in departments of entomology? So part of the research that is conducted at our laboratory is in literature reviews. This is a forgotten science, in my opinion, because many people feel that science is based upon producing tables and graphs and figures and doing all that kind of stuff, when in actuality, canvassing the literature, especially when you're trying to come up with a new theory of insect olfaction, becomes necessary. So in reviewing things, I ran across the paper from Kyle from Germany in 1984, as well as Wojtasek and Liel, Walter Liel's at UC in Davis in 1999, and both of them found, using 3 different cation markers, that there are fixed negative charges present on the dendritic membrane of the sensilla. Well, this is a useful information to start. If I have fixed negative charges that are unmoving, they're fixed, then I might be able to play around with this from an electromagnetic standpoint to see what's possible and what's impossible. So when I saw this paper, I actually had a flashback back to my senior year at Cornell University. Now, I'm not talking about some of the flashbacks that you may have when you go back to your senior year at whatever college you happen to attend, but when I was at Cornell, I did a senior project, and it involved SDSPAGE. SDSPAGE stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis, and what this process does is it separates proteins. Now, it separates proteins by what you do is you have the proteins, the process of separating them I'm not going to go into, but you then coat them with negative charges, and the more negative charges they have on them, the bigger the binding protein is, and so that's the idea of how to separate them. So what we do is we place the proteins into these little wells right here on the top, and they then fall through once we create an electrical charge. So we have a negative electrode at the top, we have a positive electrode at the bottom. They're coated with, it's a negatively charged protein, so they're going to be repelled by the negative electrode when we turn it on. They're going to be attracted to the positive electrode, and they will then move down in the straight direction, and they will then create bands. And these bands will separate the proteins according to charge, but according to our understanding of it, they're actually being separated according to size, because that's what we're looking for. So understanding how this process works and realizing the research that I just showed you before, I thought there's something that we might be able to play with here. If I've got fixed negative charges on the dendritic membrane, and the current theory says that the binding protein gets to that, and I just spent an entire presentation in 2009 saying that there's no way, using the laws of diffusion, that it's going to make it in time, could there be something else going on? I've actually spent a lot of time trying to ask people, is there something else that I'm missing? So when this idea came to me, I thought, I'm going to go with it right now, because if I got a negatively charged membrane and I can show that the binding proteins are positive, then I've now got electrostatic attraction occurring between the binding protein, which we know has to get to the dendritic membrane. Whether it separates the pheromone or not, it's not important. I just need to get that binding protein to the dendritic membrane. With my fixed negative charges, just like SDSPAGE, I may be golden. How do I do that? Well, when you analyze amino acids, or proteins, I should say, you're taking a look at 20 amino acids. There's only 20 amino acids that we need to worry about. These 20 amino acids are listed here with their 3-letter code as well as their single-letter code. And obviously, this is a lot to analyze. I don't want to analyze all of these. So what I'm going to do is I'm just going to analyze some of them. I want to analyze the 5 that have a charge associated with them. And it's those 5 that I'm going to focus on, and I'm just, for the purposes of this presentation, going to ignore the other 15. So I've got those 5. Aspartic acid, glutamic acid, both of them negative. Histidine, arginine, lysine, all 3 of them positive. Now, if you play the odds, if you like to play in Vegas, and you want to play the odds, I'm looking to get positively binding proteins. I've got 3 positives and 2 negatives, assuming an equal distribution of the amino acids. I mean, this is just a win-win situation. So I could say all I got to do is add this up, and I'm going to be able to show that our positively charged binding proteins indeed might be attracted, and therefore I could speed up unnaturally the process of diffusion. So how do you do this? You obtain the amino acid sequence for a given binding protein on the NCBI website. Next, you plug in the amino acid sequence in order to determine the signal peptide. All peptides, when they're produced in the nuclear membrane, have a little bit of extra stuff on them. And after they pass out of the nuclear membrane, before they're activated, they're cleaved. And that signal peptide is cleaved off. So I wanted to determine what the signal peptide was so that I knew what the active form of the binding protein was. Then I needed to determine the number of each charged amino acid, add up the number of positive and negative charges, simple arithmetic, and then finally calculate the net charge on the binding protein. So let's start there. I started analyzing binding proteins, and there's a lot of them out there right now. These are the pheromone binding protein charges on the Saturniid moss. The Saturniid moss are those large, big moss, the Luna moss, Cecropia, Polyphemus, and everyone likes to study them. So you take a look at the Polyphemus moth. It's got a pheromone binding protein 1, pheromone binding protein 2, pheromone binding protein 3. Continue on down the various species of Saturniids, and you could see that I'm in trouble right from the get-go. As a matter of fact, only 3 of the 16 binding proteins on this slide are positively charged. I'm going to keep going. I was like, well, there's got to be something going on here. So I keep looking. I look at other Lepidoptera, not the Saturniids. All the species listed there, there are various binding proteins, and there are more than one associated with each one. Negative, negative, negative. As a matter of fact, I've only got 2 out of 15 that are positively charged. I'm very competitive, so I keep going. I analyzed more on the Lepidoptera. As a matter of fact, we even had 2 forms of Manduca sexta, pheromone binding protein 2. I analyzed both. And I said, well, one might be right. I don't know if both will be right. Analyzed those as well, and 1 out of 16 was positively charged. I continue through the Lepidoptera. I've got 1 out of 14. I think you can see where I'm going. Obviously, the frustration is beginning, and I feel that Nobel Prize just being pulled out of my hand. So there's actually Austrinia nubilalis has a number of strains, 2 strains associated with it, different receptivity to 2 different pheromone concentrations, so I analyzed both of them. I was getting no help there either. 0 out of 10 were positive. Took a look at some more. 0 out of 16 were positive. Then I moved on to the Hymenoptera, which are the wasps, bees, and ants. And I was, not only was I not getting the positives that I was looking for, but when I moved to the Hymenoptera, we started getting some high level, negative 12, negative 14, we got negative double digits. So we're no longer dealing with what we had in the Lepidoptera, 12, 12, really highly charged, negatively charged binding proteins. The scarab beetles. Scarab beetles also had a pretty consistent high. There was one at negative 10, but they're all between negative 5 and I think negative 10, with the exception of this one, which is at 0. And we charged other groups. I took a look at Drosophila melanogaster. Why not? Everyone else does. And I had a little bit of confusion there because they don't know what the binding protein is because they really don't have a pheromone. But they have a pheromone binding protein related protein. Yeah, we love acronyms, don't we, as scientists? So I don't quite know what they are, but I analyzed them. I wasn't getting any help there. Formia regina, caliphorid fly, those that attack deer on the side of the road, those maggots. Negative 14, that's considered to be in the family. Cockroach here, negative 3, and a thrips attacking your flowers coming at negative 10. Again, I'm in trouble. I'm not doing too well. Let's just summarize. Pheromone binding protein charge summary. We analyzed over 150 binding proteins, but we only counted the proteins with a known signal peptide. If we could not calculate the signal peptide, we let it go. Of the 150 that we had, 9% of them were positive, 9% of them were neutral or 0, and 82% were negatively charged PBPs. As I said, I'm competitive. There's got to be something else going on here. Let me take a look at other binding proteins. Now, there are the pheromone binding proteins, which bind the pheromones. There's also the odorant binding proteins, which bind various odorants, like plant odors. I analyzed these too. There's a lot more of them. Over 650 binding proteins were looked at. Only the odorant binding proteins, antennal binding proteins, and the general odorant binding proteins were used in this analysis. But again, I had 66% negatively charged, 9%, same as last time, coming in at 0, and 25% coming in. I was not getting consistent results. And if I'm not getting consistent results, I can't apply this to the original idea that I had of trying to speed up the process of diffusion via electrostatic attraction. So, in a sense, you go back to the drawing board, because as a researcher, you have to do this sometimes. And you run up into roadblocks, and as researchers, I run into more roadblocks than I run into positive results. So I go back and I read the paper by Kyle. And he says, which I missed the first time, because I guess it wasn't important to me at the time. But there are fixed negative charges present on the inner cuticle of the sensillae in addition to being on the dendritic membrane. Well, this is interesting news to know. This is very interesting news to know. What does that mean? Let me show you. We now have electrostatic repulsion with my negatively charged binding proteins. We've got negative charges on the inside of the cuticle. We've got negative charges on the dendritic membrane. My binding proteins are largely negative, which means they're going to be repelled from both sides. As a matter of fact, they're going to be repelled with one another. They've got the same charges. Even if they're positively charged, they're still going to be repelled from one another because of similar charges. So I've got them being repelled from the sides. This side, that side, I've got them being repelled from one another. And so therefore, we've set up this strange situation where instead of being able to travel or diffuse across the sensillar space, I've now got all of this electrostatic repulsion going on, which is now adding a certain amount of rigidity. My fluid dynamics have changed. I'm no longer dealing with a nice, clean, flowing water, which is where all the diffusion coefficients are calculated from. It's either air or water. Those are the only two we have. Now, I've got this situation a little bit rigid, which makes sense because I was talking to a German researcher who said that the inside of the sensilla is more like a gel than it is anything else. I'm starting to agree with them now. I had that conversation a few years ago because if you do not have movement, then the pheromone binding protein is not going to be able to move to the inside. Imagine all these balloons getting in there. You're not going to be able to get through all of these balloons because of this rigidity right now, which means diffusion has been compromised, and I mean in a bad way, which means the original diffusion coefficients that I calculated for the 2009 presentation are now on a completely different scale. When I say completely different scale, I mean really, really low. For those of you involved in SDSPAGE, you know that in that gel, you will not get proteins moving through that gel unless you turn on the charges. For those of you who've dealt with SDSPAGE before, and if you've done enough of it, then you know that every once in a while, you forget to turn it on. And so you come back and you analyze the gel with chromaticity brilliant blue, and you realize, oh shoot, they're all stuck at the top. I forgot to turn it on. The reason is because proteins do not easily diffuse through a gel. Well, great. Now what do I do? There's two philosophical implications with this. Either binding occurs, the pheromone gets to the dendritic membrane, or it doesn't. One of the two. It gets there or it does not. If binding occurs, technology has just not found a way to show this yet. That's the excuse I get when I go to conferences. Binding does not occur. If that's the case, then, philosophically speaking, detection of the pheromone must occur through some alternate means. What is that alternate mean? Well, there are two theories out there, the lock and key model, which most biologists are familiar with, even non-biologists, I'm sure. And then we have the vibrational theory of odor, which was brought out in the 1930s by Dyson and has been resurrected a little bit today. And so to take a look at the vibrational theory of odor, there are certain conditions that need to be met. If we're dealing with an electromagnetic signal, then conditions must be met. I've got some if-then statements right now. Let's follow along. If this is electromagnetic, then the message from the pheromone out here needs to get across the cuticle. If this is electromagnetic, then this message, after it passes the cuticle, must be able to pass through this entire watery matrix right there, or this gel as I've just determined. If this is an electromagnetic message, then I must figure out some way in order to activate this receptor without the pheromone actually making contact with it. Those are my if-then statements, if this is occurring electromagnetically. Is there some way for me to satisfy these conditions? Let's take a look at some circumstantial evidence. There's my first, actually I forgot to put these numbers up, and there's the second region and the third region for effect. Dielectric. If this is acting as a dielectric antenna, as Phil Callahan, the mechanism that has been put forward is shown, then a dielectric, according to the definition, needs to have, di means two, electric means that we have separate electrical charges on either side of a non-conducting material. Excuse me. The conductive material passes electrons freely. So you have positive and negative charges on both sides. It's not maintained. But a dielectric material, that which is insulated, is able to maintain charges, let's say positives on this side, mostly negatives on this side, and is able to do that because of its dielectric properties. A dielectric antenna is different than a conducting antenna, although they can pick up similar frequencies. So if we're dealing with a dielectric antenna, let me hit the literature again to see what I can find. And there's not a whole lot of papers out there dealing in bioelectromagnetics of insects, but there are some. So Erickson, back in 1975, I found that the mean surface potential of honeybees varied throughout the daily flight period. If the bees are leaving the hive, they've got a surface charge between negative 14 millivolts to 8 millivolts. Take-home message, they're about 0. A little bit positive, a little bit negative. After they're flying, static charges are built up. When the bees return from the hive, they are universally positive. They're between 13 to 59 millivolts on the outside of their body when they come back from the hive after flying through the air. Take-home message here, I've got positive charges on the outside of the insect. So far, so good. That's something, at least it's consistent. Remember this slide that I talked about before? I said that there, well, not I said, but Kyle said fixed negative charges are present on the inner cuticle of the sensillae. Wonderful. I've got positives on one side. I've got negatives on the other. I was able to satisfy, and I'm not going to talk about the other properties of a dielectric antenna. Why? Because I've talked about this before at SSE meetings. But I just want to talk about this new evidence right now. I've satisfied another condition of a dielectric antenna on the actual sencilla itself. Now we've got the second part. If I'm going to have an electromagnetic signal pass through a watery matrix, how am I going to do that? Well, some of you will say, well, you can't do that through water because deionized water doesn't conduct. And I would say, yeah, you're right, it doesn't conduct. What about water? Well, water's a little more conductive. It depends, Tom, upon the various impurities that you have in there. And you're right, you can get some conductivity of water, but how can you make it more conductive? Take some salt. Take a salt shaker on top and start, throwing the salt in. And as you're putting more and more salt into the solution, you will find that it will become more conductive. So if I'm going to have a conducting fluid, do I have these salts present? In fact, I do. I mean, this is just classic biological principles. The Ringer solutions, these are all salt solutions. So we have all of the salts necessary. Calcium plus 2, magnesium, sodium, potassium, hydrogen. We've got chlorine and hydroxide ions. Those are some of the classics which are found inside. So I do have a salt solution. However, the more salt I put in, the more conductive it gets. So how much salt can I stuff in this in order to make it conductive? What additional charges can I put in here to make it more conductive than just a salt solution, which, by the way, will satisfy my conditions? What about the binding proteins? I just spent a whole bunch of slides telling you about what an incredible failure I was at determining the positive nature of the binding proteins. But can I use these to my advantage? Now I've got these charges sitting in my supposedly conducting fluid. Now some of you might say, well, I understand what you're saying, Tom, but didn't you just say some of them are 0? Yes, I did. But remember, even the ones that were 0 had negative and positive charges on them. As a matter of fact, the binding proteins are running between 30 to 60 charges per molecule, and the concentration of them is between 10 to 20 millimolar. Very high concentration, lots of charges on them. So I've got the makings of a very conductive fluid, especially if it's more of a solid. So if I get a nice matrix effect, I'm going to have a little bit of a crystalline effect, and therefore it's going to be even more conductive. So I'm feeling good about this. Let's move on to the third part. What about the protein itself? Can the protein respond to an electromagnetic field without actually being touched? What do we know about these proteins? Well, originally they were determined to be 7 transmembrane proteins. And so a classic GPCR, which was originally what we thought they were, a G protein coupled receptor. And they're all in the membrane of the phospholipid bilayer. And the odorant receptors were originally placed in the Class A or Class 1 rhodopsin-like family of GPCRs. However, they could not be placed in subfamily A1, subfamily A7, or subfamily A16. They were given their own category. They could not be placed in any. So they looked like they were in the Class A. Rhodopsin, by the way, just to remind you, responds to electromagnetic energy. But it looked like it was close to this, but they were placed separately. They were not actually called subfamily A20 because they weren't too sure about it, but they were placed there. So this is what it looks like. We've got a 7-transmembrane molecule. This is what all of them look like. There is no variability. Just in the past couple years, analysis of the odorant, the odorant receptors have shown that it may be a 7-transmembrane protein, but it's backwards. All GPCRs have the amino end outside, the carbon end on the inside. The N's and the C's were switched. All of the odorant receptors in insects are backwards. Everyone, even as I'm standing before you, for those who are watching this video right now, are still scratching their heads, wondering why the insects have just made this exception. They are the only ones with a backwards odorant receptor. So I get attacked. Sometimes this happens. And one of them is that, Tom, you're wrong, because we've been able to show that the lock and key is valid because we deal with xenopus oocytes. For you non-biologists, it's frog eggs. So what they do, there's our classic frog right there, xenopus laevis. And they use these a lot in biological laboratories. And those are the eggs off to the right. And what they do is they express proteins in these xenopus oocytes, and then they analyze them. It's a rather simple, it's an elegant system. What they do is they inject it, I think it's with cRNA, and it takes about 2 or 3 days for them to express the proteins in the actual membrane of the frog egg. And then you start testing the frog egg. You actually start throwing odorants at it and looking to see if it responds. It's really kind of a neat system. So they were able to do that. They were able to take the odorant receptors. Now, they did have to have the dimer because there are 2 proteins involved. There's an OR83B, which is a co-receptor, and similar to it, which have just recently been called ORCOs for odorant receptor co-receptors. And these ORCOs, in addition to being expressed with the odorant receptor, now give a certain amount of activity to the frog eggs when you hit them with an odorant. Therefore, Tom, you are wrong. Because we've got the odorant, we put it in this artificial system, and we get a response from a frog egg. And all we're doing is expressing the protein. And that is some convincing evidence, to be sure. And so I turned to some of the individuals and I said, well, did you establish contact? And he said, well, what do you mean contact? I said, did you establish contact between the pheromone and the receptor? I understand it was expressed. I get that. I understand you exposed the odorant, the appropriate odorant. I get that too. Did you show contact between the pheromone and the... Well, yeah. I mean, how else did it happen? I mean, we put it there. We got a reaction, a depolarization. So, of course, it contacted. I said, what process did you use to show contact? How do you know that they made contact? One of the persons got frustrated at me and said, how do you define contact? Touching? Follow along. My 2 hands are separated. I bring them together. They're now touching. That, to me, is contact. That's how I define contact. Was contact shown? That conversation ended, by the way. So no contact was shown. But what was shown, even though it's not the same reaction, we're not getting clear spikes, we did have activation. The odorant and the odorant receptor, we got activation in the frog oocytes. So how was it being activated? We're not sure if contact was made. It might have been. As I'm standing here right now in 2012, it might have been, but it just wasn't shown. And by the way, this system only worked in a salt solution. Moving on to another study that I found. Take a look at what the Japanese researchers did back in 1985. Evidence for non-receptor odor discrimination using neuroblastoma cells as a model for olfactory cells. So we've got a model situation, a very unnatural situation. The excerpt from the abstract, the mouse neuroblastoma cell, which is independent of an olfactory cell, was depolarized by 20 different odorants examined, suggesting that specific proteins are not required for reception of odorants. Well, this is kind of big, because lock and key says that if you've got an odorant, you need to have a very, very specific odorant receptor. It's lock and key after all. And because it's lock and key, it has to be specific. I mean, you change one small thing on an insect pheromone, and you will change its receptivity forever. So it is very critical to get the right one. Or is it? Let's get even a little bit simpler. The frog eggs is a living entity. But what if we can simplify the system even more? So here we have a phospholipid bilayer. The hydrophobic end's on the inside, the hydrophilic end's on the outside. Most of you, I think, are aware of how all membranes are set up. So we can make these called liposomes, and what these are is these are just a phospholipid bilayer, and that's it. What do you mean that's it, Tom? Is there anything inside? No. Is there DNA? No. Is there RNA? No. Is there a nuclear membrane? No, there's not that either. What about endoplasmic reticulum? No. Mitochondria? No. Is there anything inside? No, there's nothing inside. It's just a phospholipid bilayer. So what do you do with it? I'll tell you. Here's a phospholipid bilayer. I've blown it up right now. We've got the two layers right there, and you can insert membrane proteins into it. Well, this is a nice, simple system. At least I don't have to deal with a frog right now. So we get these liposomes. You insert the membrane protein in there, and you can also play around with them. So what do we have? If we insert a membrane-bound protein into a liposome, we create what is known as a ProDo Liposome. I think you can understand the word derivation for that. So if we have an insect OR, and we then place, through means, all of these membrane-bound proteins into it, we can now test the liposome for its activity to different odorants. A much simpler system than even the frog eggs. What do we get? We get activation. So we express it in a liposome, and we get activation, which is a type of depolarization. It's a subtle signal, not quite the same by far as an in vivo situation, but we're getting a reaction from it. That's telling. What about a random protein? Just got finished with that 1985 paper, said they weren't even using the right odorant receptors and they got response. What if I had a random protein? What would happen if I took a random protein and then put that in the liposome and then tested an odorant that we would assume is nowhere near related to it? What would happen? We would get a reaction as well. Why? If it's lock and key, what's going on? Are these odorants, are they locking into every single protein that we give it? Nomura and Kurihara in 1987, taking a look at liposomes as a model for olfactory cells. Various odorants were found to depolarize, and these are particular type of liposomes, as lectin liposomes. Beautiful, interesting stuff. The lipid composition of the liposomes is one of the factors that controls the sensitivity to the odorants. That makes sense too. You change the phospholipid bilayer, you change the properties. 3, odorants change the membrane fluidity of the liposomes. What does that mean? The membrane fluidity is that it has become destructured, shall we say. Why? This is actually important, because anytime you depolarize something, you cause leakage. Leakage? What do you mean leakage, Tom? You mean cell is leaking? Yes, that's how all depolarizations occur. Ions leak into the cell through an ion channel. This leakage is necessary, and part of this leakage is dependent upon destabilizing the membrane. This is actually one of the prerequisites, and was known back in '87, is that when you change the membrane fluidity of the liposomes, you are now creating a sort of activation. And finally, the membrane fluidity was changed in concentration ranges of odorants similar to those where the membrane potential changes in a regular in vivo situation. So it turns out that this is relatively accurate, not too bad to work with. 2 years later, the same guys came up with another study. The responses to odorants in these liposomes diminished at low concentrations of salts. and recovered upon addition of salts to the stimulating solution. I mentioned that salts were necessary for the frog eggs. This didn't really ruffle any feathers because people figure, well, it's a frog egg. It needs to have some sort of ringer solution for it to work, but the liposomes don't need that. I mean, it's essentially just a dead phospholipid bilayer. So you take away the salts, you lose the effect. Why? If it's lock and key, what would the salts have to do with it? Why does the addition of salts bring it back as soon as you remove them? They also found that divalent cations, divalent meaning 2, were effective in supporting the responses to odorants at much lower concentrations than monovalent cations, 1. 1 positive charge versus 2 positive charge. You see here, the divalent cations supported it at much lower concentrations. They increased the sensitivity. So when you increased the charges of the in vitro situation, you increased its ability to detect the odorant. The next step was obviously, well, we've got ions going in and out. I mean, the whole leakage thing, you know, calcium influx, this is how neurobiology works. What if we used impermeable organic cations? So we got those charges, which I'm suggesting are important, but they're impermeable. So they can't get inside the liposome, which means they can't be causing the depolarization effect. When they used impermeable organic cations, they were effective in supporting the responses as well. So the response is not based upon an influx of calcium ions. You simply need to have the charges on the outside in order for the system to work. And let's see, this one is 1990. Again, Japanese researchers, I guess they're playing a lot with liposomes over there in Japan. Need to talk to you about that. We've got some proteins right there, and they were able to show this effect, as I told you before. But what if we took some of those same proteins and got a little more complicated? What, instead of having one odorant receptor, we had a whole bunch of different proteins? What happens then? They actually found out, the Japanese researchers, that the more complex you get, the better they get at detecting odorants. How good? In this study, they used 12 different odorants. The liposome, this dead molecule, this, I don't even know what you want to call it. Let's just call it a liposome. This liposome, this non-living entity, is responding to 12 different odorants that it hit it, and they were consistent. Odorant number 3 was giving exactly the same response every single time, to the point where at the end of the study, they could tell which of the 12 odorants it was based upon the response that they got to the liposome. And to answer your unasked question, no. None of these proteins that were used were classic odorant receptors, just random proteins. Well, this then moves on to some other possibilities. I mean, indeed, if this conducting fluid is important, then where to go from here? So I got a chance to talk to a researcher at Georgia who does work on decapods. Decapods are lobsters, crabs, shrimp, 10 legs, crustaceans. And I was chatting with them one time about the sensory cells, the aethotasks. And they mentioned to me that there's a way in order to make them anosmic, which means they can't smell. And I said, well, how does that work? He goes, you flush them with fresh water. And I thought, and I said, fresh water? You're kidding. And I said, does anybody else know about this? You know, think it was like some big secret. He goes, oh yeah, we all do it. You all do it? So you flush the lobster with fresh water and it doesn't smell anymore. He goes, yeah. And I said, okay. How long does it take? And he said, well, at least 45 minutes. So we do that to kind of clear them out. It's kind of like a control type thing. And then once the saline solution, because lobsters live in the ocean and marine environment, the salt solution gets back in there and everything is all fine. And I thought, really? That's interesting. So I got a chance to talk to another researcher also dealing with this issue at the University of Florida. And we were chatting about blue crabs, because blue crabs are able to survive in a marine environment, salt, and a freshwater environment, because they migrate. They can do both. So I said, well, what happens with the sencillary cells? And again, just very quick, off the cuff. He goes, well, they've got this salt solution that they kind of move in and out of the cell, and the sensory cell at will. So as they're moving from the marine environment up through the Great Lakes, for example, the actual change of the microenvironment inside the sensory cells is actively changed by the blue crab. And there's Gleason's paper in 2000 attesting to that effect. And I thought, wow, this is really interesting too. I mean, I'm an insect guy. I don't usually deal with decoupage, but in this case, I'll take a look at this because there are some similarities and differences between the decoupage sensory cells and the insect sensory cells. Well, taking this idea then, we should be able to come to some conclusions. I mean, if this salt solution is important, supposedly as a conducting fluid, then we should be able to come up with some if-then statements. If this is true, then terrestrial insects need to have this salt solution. Therefore, we've got the pores, and there shouldn't be a problem because it's in air. Therefore, I would expect them to have the pores and that they would have this microenvironment of the salts, and they do. They do have pores in the olfactory sensillae. Crustaceans, what about crustaceans? Can they have the pores? Some might say, well, no, because everything would be leaking. I was like, well, it really wouldn't be that bad because the crustaceans are in a marine environment, and if they're in a marine environment, the salt solution, as it's getting in through the pores, is still going to be able to allow this to occur. That's not going to be a problem at all. So they do have pores in the olfactory sensillae. What about aquatic insects? Insects are not found in a marine environment. They're in fresh water. What then? There's a curious thing with aquatic insects. I had to go searching for this. No one really knows why, but aquatic insects do not have pores in their sensilla. It's just not there. So therefore, they're able to maintain that microenvironment. And these same aquatic insects, once they come out of the water, like dragonflies, stoneflies, mayflies, they come out and they're flying around, they get those pores back. So this is what's going on. This is what's going on. There is some suggestion right now, some circumstantial evidence, new evidence that I'm supporting right now to show that indeed we may have this system actually occurring, an electromagnetic detection based upon sound physical laws that could be occurring in the insects. And with that, I am done. Oops.
Time for a few questions.
Roger, can you put the microphone over there? Thank you. This picture was also taken a couple days ago. She sent me 2 pictures. I had to choose. I couldn't choose. Go ahead.
Nice picture. My name is Larissa and I want you to elaborate a little bit on the possibility that the olfactory mechanism is based on a quantum effect because there are not enough protein conformational changes to explain the large number of smells we can recognize, at least in human olfactory system.
Yes, that's actually going to be outside my area of expertise. She's actually bringing up a point, I think, about the electron tunneling theory that Luca Turin has come up with. There is more of a physical effect. It does not seem to be occurring with our system because with his effect, you have to be between 2 plates for electron tunneling to occur, and it's through this process, and it is a quantum type of effect. Whereas with this system, because it's occurring at a distance, the evidence to me seems to be more electromagnetic than a pure quantum effect. So I would differentiate between the 2 theories and suggest, because I'm not able to elaborate mostly, so I'm trying to dodge the question, is that I would think it would be more electromagnetic, and therefore we're probably dealing with more of a wave phenomena than we are with the quantum type of phenomena. Although, I still could be wrong. The jury's still out. Thank you.
York Dobbins here. You've been challenging the paradigm for insect olfaction for quite some time, and in this talk you just pointed out that there's evidence for the same thing going on in other arthropods. Do you think that there might be a similar challenge to the lock and key model of olfaction possible elsewhere in the animal kingdom?
Absolutely. As a matter of fact, am I able to stop my answer with just that one word? Because obviously I don't have enough time to go into it, but York, Thank you for the question. Yes. The answer is yes.
Glenn Ryan, and the question, well, first a comment, are you aware of Ross 80's work back in the 80s where he demonstrated that electromagnetic fields affect membrane fluidity in neuronal cells? And are there other similar studies that have been done with olfaction using physical electromagnetic fields to alter the processes you've talked about?
Well, actually, you've just brought one up right now. But to answer your question for insects, no. Yeah, for insects, no, but Ross's research and others, yes. I mean, you can play games with electromagnetics and get the responses. However, this is usually kind of scoffed at. We're like, well, you know, of course there's membrane potentials, and you're probably just doing something really weird to it, something unnatural. Whereas you and I might be saying, oh, no, I don't think this is unnatural at all. I think there may actually be something to it. But absolutely, yes, I am aware that there is other research out there, but I tend, and this may be a mistake of mine, I tend to stick with the entomological research. Why? Because it's more convincing to the entomologist. So if you go too far away from bugs, people catch you and say, well, you know, I don't read that literature, so essentially you're discounted and you really can't go anywhere. But to bring in decapod crustaceans, because they're so close, they're the arthropods, they've got many strong similarities between the sensillae of insects and the aesthetasks of the decapod crustaceans, then I think that's a valid comparison to make, and it was a welcome one, too, because I didn't expect the similarity to occur. It just kind of fell on my lap. I decided to go with it, and that's why I threw up a couple slides for your benefit and for the benefit of those who are taking a look at this video right now.
Thank you, Tom.
Thank you, Roger.
Thank you, Doc.